Hello, everyone. Good afternoon. I hope you can all hear me online. So welcome to the third of our Director's Choice Seminar series. Today, John Upham and I welcome you to talk about biobanking, which is a very complicated area, and we'll be discussing the needs of biobanking and how it can relate to clinicians and biomedical researchers. I'd like to begin by acknowledging the traditional owners and custodians of the land and pay my respects to elders past, present and emerging of the Yagara and Turrbal people. In this seminar, we will be exploring the enablers and the barriers to biobanking and ensuring that those samples are available to our research community. I'd like to start by welcoming our first speaker, who is online, Associate Professor Andrew Moore, who is based at the Queensland Children's Hospital, where he is a senior staff specialist in Paediatric Oncology and Haematology, and the Director of Research at the Queensland Children's Hospital. He is also Director of the Queensland Children's Biobank, and has a number of national roles, including being on the board of ANCHOG, and he's also the PI of several large Australian paediatric clinical trials in oncology. So I give you Andrew Moore. Thank you.
Good morning. Hi, thanks very much, Maher. And my apologies that I can't be there in person this morning. I had a clinic this morning and taking over on call immediately after this meeting. I'll try and share my screen. It's not letting me at the moment. It just says can't share screen while the other participant is sharing. Of being the director of the Queensland Children's Tumour Bank, which is administrated by the University of Queensland. So I'm presenting with my UQ academic hat on today. So the Queensland Children's Tumour Bank was the second paediatric tumour bank in Australia, behind that at Children's Hospital Westmead. Is established by Andrew Hallahan back in 2008, and the directorship passed to me in 2013 when I returned from the UK. We currently have 3 FTE of staff. The engine room of the tumour bank is really led by Melissa Maybury, our tumour bank coordinator, who's on leave. Otherwise, she'd be present there at TRI for the talk this morning. T-Fam has been with us for several years in a senior technician role, and I don't have a bio because he's only just started, but Clark Vincent recently joined us from Maher Pathology as a research technician. So I'm very fortunate to have 3 excellent staff, and it's very much a close collaboration between the UQ Child Health Research Centre, which is based here on the Children's Health Precinct, the Oncology Services Group at Children's Health Queensland, and also Pathology Queensland. And all 3 are co-located in the Centre for Children's Health Research building where I'm presenting from, which is across Raymond Terrace from the Queensland Children's Hospital. So it's a very strategically located tumour bank in a strategically built building. We're part of the ANCHOG Biobanking network, so the Australian and New Zealand Children's Haematology Oncology Group. I happen to be the director of that biobanking network, which is funded by the Kids Cancer Project. That funding supports a network coordinator that really aims to link all of the paediatric oncology tumour banks around the country. These are either placed at research institutes or within the clinical service at each of the ANCHOG Children's Cancer Centres. And the idea being is that if one centre has a collaborative research opportunity, then we reach out to the other banks and leverage the number of samples that are around the country and ship those to, say, an international collaborator, given the rarity of the tumours that we deal with in general. In terms of the overall principle, none of this will really be of earth-shattering news. The idea behind biobanking is really to bypass the bin. Obviously, patients are consented for any excess material that's not required for diagnostic purposes at their point of biopsy or definitive surgical resection, or both, as the case is for our tumour bank. The diagnosis is obviously made at pathology, and then the patients begin their treatment journey. Normally, the excess material is discarded, and so it's really important that we capture that material and make it available for research. And we do that in-house here at the QCTB. Our consent is a very seamless process. A range of clinicians are able to take consent. It's on the QEP's website. So whether it's a surgical registrar or our oncology registrars or oncology consultants, they all have the up-to-date consent form and can enrol patients. We also actually have ethics to have receipt and processing and quarantining of samples from patients that might have had an unexpected diagnosis of cancer. So a classic scenario might be the patient that has a laparotomy for a bowel obstruction, an intussusception that turns out to be Burkitt's. Happens on a weekend. The pathologist receives the sample, and it's, well, this is cancer. Obviously, the parents haven't been sort of necessarily aware that that was a potential diagnosis, and so it just saves a lot of logistical running around if that we can access and process that excess sample. And particularly given that we preserve it in a cryopreserved way, we then get consent from the parents once the oncology diagnosis has been communicated through to them. And clearly, if consent's not forthcoming, then that sample is discarded. And so the process is very seamless, with Pathology Queensland as a key partner, and we collect all manner of tumour-related tissues and fluids, as well as post-mortem donations on occasion as well. I'll talk a little bit more about that. Our processing and de-identification has a very clear SOP and process, and our samples are cryopreserved. In terms of, we are able to collect at multiple time points from diagnosis, relapse, and as well as germline material for comparative analyses from our patients. Our recruitment has really increased year on year, and there's been a number of key facilitators of that increased recruitment. The biggest one of all, though, is the return direct benefit to patients, and we achieve that benefit to patients by a close partnership with the clinical service. I'm part of that clinical service myself, but a number of our clinical trials, which is really the standard of care and approach to managing childhood cancer, require tissue to be sent to the Central Clinical Trials Unit for correlative biology studies. So you can't be enrolled on the therapeutic study if you don't have a biological sample. And a number of our academic trials also feed back directly to patients. So it's proven a very successful program over the years, particularly when the clinicians understand that there's a direct benefit to the patients. And clearly, we also have a much bigger contribution in the research space. So we've so far enrolled over 1,600 patients since the Tumour Bank opened about 13 years ago. And the number of specimens collected per year has been increasing as well, hence the slightly increased staffing that we've had to have in recent years. And some over 4,600 events we've collected, and many-fold more vials than that. In terms of our solid tumour processing and future-proofing, in very simple terms, the pathology will place the representative tumour material into tissue culture media that we receive fresh and straight away. We process that in various formats in terms of processing snap frozen pieces of tissue, The pathologists work with us very well so that we know which corresponding sort of block from the histopathology we're dealing with to ensure that it is tumour material that we've got. So we have snap frozen for genomic analyses and molecular analyses. We preserve a cryo-mince, which is essentially a minced up part of the tissue in tissue culture media, which allows functional studies in future. And then depending on the collaborative nature of the project and the tumour type, some of the material, if there's enough, will also go straight into the hands of local collaborators for xenografting or in vitro work. So it's a very, it can be a very time-consuming process, but given really good collaboration with the clinical service, it is fairly smooth. Similarly, for our bone marrow aspirates and blood, we FICOL prep to enrich for the mononuclear cell layer. And again, we split these samples into snap frozen cell pellets, as well as cryopreserved mononuclear cells. We marry that up with the blast percentage by flow cytometry from diagnosis, so that we know what the leukemic portion of the sample is in each vial. So it's just a picture of Mel and T at work. We've got a dedicated tissue culture hood and an area in the lab here at CCHR, so we're not competing for space. We've got our own bench areas, and as well as the lion's share of the cryopreservation tanks in our own facility for -80. We use the REDCap database, which is a bespoke solution that Mel has really developed over a number of years that works very well to track the samples as well as the data associated with those samples. And most importantly, our tumour bank is there to drive translational research. We've had a number of collaborative projects looking at novel markers for leukemia, as well as novel MRD assays and improving diagnostics with genomics in particular. We managed to translate that into a Queensland genomics project with the Archer platform. As well as from a solid tumour perspective, we've had a number of key collaborations with international partners and some 56 collaborations worldwide in the 13 years of the tumour bank, which we're very fortunate and proud of. A number of these have really been quite game changing in terms of leukemia, for example. Charles Mulligan's work we contributed to in the characterisation of pH-like ALL. And indeed, the patient that we contributed to had a direct benefit from this, was worked up in real time for a patient with refractory disease, and identified a kinase target and commenced on dasatinib with response. So again, it really speaks to the power of our tumour bank experience in that we've been able to contribute both to global collaborations, but with a circular loop back to direct patient benefit, which has been really quite compelling. In addition to that, just highlighting some of our big papers that we've contributed to. Again, you'll note the journals are at the upper echelons of academia, which is always exciting to be part of. But more importantly is the stories that these tumours contribute to. When you pool samples from across the world, particularly in rare cancers, that's when you really make genuine discoveries. And in terms of CNS PNET, for example, there's a myriad of new molecular subtypes that were just not previously known due to sample size limitations, as well as obviously the advent of genomic analyses. Similarly with medulloblastoma, what we thought was 4 diseases is more likely 12. Based on the subtypes. Again, global collaborations and contributions from our sample sets. We've had an excellent relationship with Michael Taylor's group in Toronto. And then more recently in the Australian space, the collaboration as part of the Zero Childhood Cancer program led out of Sydney, with whole genome and transcriptome and methylome profiling of high-risk cancers that was published in Nature Medicine last year that showed that the majority of these samples have somatic variants that are pathogenic. Many of them are potentially targetable. And so that's something that has been, again, part of direct patient care and that return loop. In terms of our oncology service, as I mentioned earlier, we really contribute and facilitate enrolment of patients on clinical trials, and that's absolutely underpinned the success and the sustainability of our tumour bank. Just by way of example, some of those are in the brain tumour space, a rare cancer space in the Zero program, and my own AML trial that we lead at a national level as part of an international effort. The My Child study, given the well-organised and functional nature of our tumour bank, in addition to having centralised flow MRD, every child with AML in Australia, for example, has their diagnostic and follow-up marrows all shipped to the Royal Brisbane and Women's for centralised flow MRD analysis. And then the samples come across to our tumour bank here on the regular courier that runs between the Pathology Queensland lab here at CCHR and at Hurston. The samples come straight upstairs to our lab. We process the samples for molecular MRD that gets shipped to the research lab in the UK, and excess material gets biobanked in our bank. So that's the other big message as part of my talk in terms of what can you do if you're setting up a biobank, is really look to leverage opportunities on clinical trials, spend the time investing in how to make sample movement work efficiently and effectively. In addition, we contribute to diffuse intrinsic pontine glioma registry for the handful of tumours that we receive post-mortem, which is an incredibly powerful and important process for families in particular. And then we also have, we've contributed to a number of studies where we can do functional assays for leukemia as well as solid tumours because of the cryopreserved nature of the samples. As mentioned, that direct clinical benefit, over 220 patients, we capture this with very clear intent and importance, because it's a key metric that we report on, and it's a compelling story for our funders as well as our clinical collaborators. Roughly a third of the patients that we biobank derive direct benefit from our activities, be that through clinical trial enrolment or molecular MRD testing, for example, in ALL, and some over 220 patients in the life of the tumour bank. So in terms of the brief that I was given to finish with the big picture learnings of our tumour bank, the key message in terms of why we've been successful and also sustainable, and that's key, is to be integrated with a clinical service. Don't just be a research endeavour. The partnership for us has a very clear mutual benefit from us as an academic research endeavour, but also the patients and the clinical service. We provide a service that is beyond the scope at the moment of Pathology Queensland. There are some discussions around PQ's role in the biobanking space, but we also have that direct benefit to patients, which proves absolutely invaluable, and is nothing gets clinician engagement more than this benefit to your patient. I'd like to think we've collaborated widely and networked widely and effectively. Our collaborators in the UK, the US, Canada, in interstate and locally will often report that our samples are of high quality, our data and our service. You're really providing a service as a tumour bank. You're a resource, and the better you provide that service, then the better the whole endeavour is. And critically, we've had excellent support from the Children's Hospital Foundation and maintaining strong engagement with your funders, which is key for any research endeavour. But it's a relationship that you need to foster and maintain over many years, because we've been fortunate to have that support, and we do many tours and fundraising activities with the Children's Hospital Foundation, and it's obviously reap rewards for everyone involved. So that would be my key big picture learnings. And finally, I'd just like to acknowledge the staff at Children's Health Queensland, particularly in the oncology service, the number of clinicians, as well as obviously the surgical teams, the haematology team, our palliative care team as well, particularly for post-mortem donations, which are really driven by parents, and an incredibly powerful thing to do. And also our key collaborators at Pathology Queensland, and of course, all the funding from Children's Hospital Foundation. So thank you. I'll stop sharing.
Okay, thank you, Andy. John Upham here, the co-chair with Maher. Thanks so much. It's great to hear what's happening through the Biobank. And I guess what stood out for me is the way that the service has developed over a period of several years, some of the key learnings that you've had, and particularly what you said to us about the integration with the clinical service and the mutual benefit for clinicians and patients and families as well. There probably will be some questions for Andy, but I'll get you just to hold those because we're going to have a panel discussion after our next speaker, and so we'll have the opportunity to talk about or ask questions of each of the presenters and the panel members. So we're just going to switch gear a little bit at this point, and I'd like to invite Associate Professor Glenda Gobe, who will be talking to us about biobanking in chronic kidney disease. Many of you all know Glenda, who's an internationally renowned kidney disease researcher. She's a research director of the Centre for Kidney Disease Research Collaboration at Princess Alexandra Hospital and co-leader of the University of Queensland Kidney Cancer Research Group. In addition, Glenda's director of the Kidney Cancer Biobank at PA, and of a new biobank which is being set up under the auspices of an NHMRC-funded Centre of Research Excellence called the CKD Biobank at the TRI. So over to you, Glenda.
Yeah, that's right. Maybe we could just go straight to this. And I just use the arrows up and down. Okay, is it on? Okay, cool. Perfect, thanks. Right. Thank you. Afternoon, everybody. I'm going to talk about two biobanks, the chronic kidney disease biobank and the kidney cancer biobank. They are not as well established as Andy's children's biobank. And so I think I can interest you in the way it sort of they develop and the problems of developing, the barriers, the enablers with developing biobanks. Okay, so the objectives are here that we wanted to get some comprehensive, we wanted to work with the clinicians. So I'm basically a biomedical researcher with a science background, very proud of it. But I know that here we have to work closely with the clinicians. So we wanted to develop a couple of biobanks where we could do that and start to get some depth and translation into our work. So we wanted to develop a couple of comprehensive biobanks that would bridge the gap between basic science and clinical research and be able to develop our knowledge with that. And also eventually to facilitate personalised medicine for patients with kidney cancer and chronic kidney disease. The operational objectives were to organise and manage the samples. So that's actually not as easy as it sounds. We needed to find infrastructure for maintaining quality samples, and then also involve people in education through this work. So it usually starts off, the need for biobanks usually starts off with a couple of projects or with ideas of projects. So with the kidney cancer biobank, we started off working with Dr. Simon Wood from urology. He had a clinical problem and we were to help him with that. And that clinical problem was that there was too much surgery for kidney cancers of a small size. And the reason for the surgery is that we couldn't determine what those samples or what the cancers were. And so the safe way was simply to remove them. And so the idea was that we would collect samples from the nephrectomies, from the cancers, save them, and then analyse them in different ways to see if we could find biomarkers or spectroscopy markers that would differentiate the subtypes of the kidney cancer. And then we would apply that, we would translate that back to patients in a scanner, for example. As you can, I think you can see a lot better than I can, the choice of treatment for most of the kidney cancers is surgery, a nephron-sparing surgery or simply nephrectomies. And so that was the idea, was to find the cancers that were indolent and then just monitor those cancers without the cost of surgery, without the changes that occur in the kidneys that are left that can then be some sort of chronic disease as well. And it was these others, the rarer cancers, here you can see they're quite a small proportion of the total of the kidney tumours. Most are renal cell carcinomas, but these rarer ones were the ones that we wanted to identify, and also the benign that definitely could stay in situ. And so we started collecting the samples and storing them in the biobank safely, collecting bloods, and started to analyse, and with good effect. In fact, we've just got a paper for revision from Nature Reports on this work. The chronic kidney disease biobank was set up slightly differently. We wanted to look at chronic kidney disease progression. Again, we had a huge database of CKD patients, over 4,000, but this is a lesson in itself. That database had never been set up so that the patient samples were collected. It was simply clinical data. And so the idea was that we would start to collect clinical samples, look at the clinical data, and see if there were ways that we could identify progressors of CKD early, and then be able to treat those patients early so that they might not progress with CKD. Or some people with CKD simply don't progress. We could identify those people as well. And so that was the initial idea of the chronic kidney disease biobank, that we would start to collect samples. We had plenty of clinical data, but not the samples. So barriers and enablers, there are plenty when you're trying to set up biobanks. The first is making sure that you have adequate ethics to do this work. We had two ethics committees to work with, North and South, Metro North and Metro South. We had to get university approvals, often forgotten. We then had 3 sites that we had to get SSAs for. And for a PhD student trying to do the biomarker study for chronic kidney disease progression, it took over a year, in fact, for one of our students to get the approval and then the site approval for this study. And that's a long time out of a 3-year project. So think about that. You need to think through the ethics very carefully too, because modifications take time. You need to think through the patient consent form, because any changes there are really important. One of the things we're starting to think about is commercialization of some of the data from the kidney cancer work, and that's not built into the PICF. And so, you know, it's that sort of consideration that you need to get right from the start. And then the identification of the people as patients as well. Yeah, I'll keep going. What are we up to? Ethics. And then infrastructure, which seems like a small thing, but owning -80 freezers, positioning them, getting racks, getting a way of digitizing the sample collection, it's really costly and not always very well supported, and is obviously something special. Then you also need the facilities for histology, genomics, etc., etc. You need a secure server. You need staff and students, and if you don't have those, you really can't get these projects going, and funding. The research governance is really important. I've taken the flowchart there from a different biobank, but we're just really setting that up now because we've had a request from a group in Sydney for some kidney cancer specimens, and so we're working through the governance for that. But the other thing that's really very important is that has been a topic here, as well, is to involve the community in where you get the biobanking with all of the governance that you need. Then you get a researcher who makes a request. It needs to go back to the to ethical considerations, but also involving the community, if you can, in this project that's a new project about to involve some of the tissues that you're using. The other consideration is having the support of the clinicians. We had, for the CKD work, we had one site. We had site approval for both of these sites. We had one site that was really enthusiastic. It was helpful. The nursing staff were just fabulous. They facilitated the collection of the tissue from a defined number of patients, and we've now finished that project and published the work. That was one student's PhD work. Site B, and the number of patients there was about 130. Site B, we're still at 2 patients, despite the ethics approval being from the same era. It's a more complicated setup than Site A, but the problem was, despite many meetings, we could never align the interests of the researchers, the development of the biobank, and the input of clinicians at this site. They'd be enthusiastic at a meeting and then it would just die away. So you really need to get input, and I suspect you really need to get a project that, you know, runs from the heart, like the kids projects. Adult CKD, sometimes it's thought to be a little bit ho-hum. The transport of the specimens as well. At Site A, they transported the specimens with other pathology specimens to PA. To transport specimens from Site B to TRI was going to actually be a huge cost. It's something to consider when you're thinking about setting these up. Then there's the basic processing like Andy had there. There's the consenting, to think about the consent for patients so that you can recontact them if you need extra specimens. And then there's the time and input for all those biospecimens that you want to collect and make the most of. And these are the ones that we typically collect here. The other, right down the bottom there where there's an NB on the healthy controls, think about the healthy controls that you need for your biobank projects and think about where you're going to find those healthy controls and how you're going to facilitate the collection of the tissue. And we had one project where we didn't do this very well. We used healthy controls from TRI with ethics approval, but they weren't considered well age and gender matched to our other project that we were carrying out. And so we published in a very low impact factor journal for that one. So think about your healthy controls. And then what about the future? You have your samples in storage, you've got your data in a secure site, and the patients have given samples to be used in research, which is pretty broad. But the problem is that this is often running with a project, like a PhD project or a grant. What if the funding runs out? What if the students and staff move on? What are we going to do then with this defined biobank? And the donors do need to have a say in what happens to their specimen. So if it's the end of a biobank, horror, reuse potential, well, of course, there's got to be. In a well-curated biobank, there has got to be reuse potential. But if it really is the end, you may need to destroy biobank biospecimens and clinical data associated with the biobank. We have decided that the biobank material will only be destroyed when there's no scientific value to be gained from stored biospecimens and clinical data. And if in the event of closure of the kidney disease biobanks and there's no entity can be found to curate the collection. It's not a small issue. The other thing that may happen with the end is, of course, if a donor requests that his or her samples are removed, and that's a loss for the biobank. So finally, barriers and enablers. The development of a biobank for a specific project or enabling collaboration with other units internationally or nationally is very worthwhile. Definitely the support of future research is worthwhile. Funding is necessary. It's very difficult to get even big institutions like the TRI, the PA. It's very difficult to get enthusiasm for a lot of money being put into biobanking and the infrastructure that's needed for biobanking. But potentially it allows research translation and the development of precision medicine for kidney cancer patients and for kidney disease patients in general. But always consider the end. There's a couple of publications there that we have been successful in putting out. The top one there, if you're interested in the development of a biobank and in the processing, just the general processing of biobanks, is by a now PhD graduate, Evan Owens, and that's the setting up of an Australian chronic disease biobank to support future research. But we have published quite a number of papers now from our biobanks. So thank you for listening.
So thank you, Glenda. I might ask everyone to come up to the chairs at the front. So together with our two speakers, we've got three additional experts in this area who'll be joining us for a panel discussion. So to begin with, we have Vicki Clifton, who will be known to many of you. Vicki's an NHMRC Senior Research Fellow involved with Maher research, one of the program leaders of the Mothers, Babies and Women's Health Translation theme, and leads the Queensland Family Cohort study. Gerald Holtman, who's a clinical academic in gastroenterology and hepatology, an Associate Dean Clinical for both health faculties, also Director of the Department of Gastroenterology and Hepatology here at PA Hospital. And then we have Rebecca Rogers, who's just getting mic'd up, who's the Pathology and Clinical Research Coordinator at Maher Research and plays a key role in managing the breast cancer biobank. So I'm thinking that we'll open it up for questions, but before we do that, we've got some real experts sitting out the front. So I thought we might get the three of you just to give us in 2 minutes a very quick snapshot of your biobank and what's happening in relation to the biobank, and then we'll open it up for general discussion. So Vicki, you're down on the far left. Do you want to start out and tell us about what you're doing through the cohort?
Yeah, thanks, John. I've started to put together a perinatal biobank as part of the Queensland Family Cohort study. That commenced in 2018. During our pilot period, we've had COVID, which changed the nature of our sample collection. And we started to transition into samples that we could collect from our families that could be posted in. And that's opened up a new discussion and thought around the fact that we are storing, currently we have over 1 million samples in storage from 500 families. And it does raise the question about, can we start to collect samples that don't need to be stored at -80? And we're thinking about collecting samples that can be stored at room temperature and samples that can be lyophilized and stored at 4 degrees. So our biobank is changing over time to accommodate for the fact that it's costly to store samples at -80. It takes a very long time to divvy those samples out, and we want to be able to store samples long term so that as we follow those families up, we can go back to samples that we've been able to store at room temperature, so or 4 degrees, rather than having samples that we keep at -80 continuously.
Okay, thanks, Vicki. Rebecca, do you just want to tell us briefly about your biobank?
Sure. So we have 2 collections. One is based around breast cancer. The other is based around gynae oncology samples. I'm actually embedded in anatomical pathology, which makes sample collection so much easier. We have a very strong clinical support base across both streams, and we can collect samples. While we do try and send the majority of our samples out fresh, we do have the ability to obviously store at -80, but also then we can then process those into FFPE blocks. We have a little bit more scope in that respect. Our clinical follow-up, I think, is our other major strength in terms of we can actually follow these patients up all the way, to 10 years post-diagnosis. So we have a huge information pool that backs up our clinical samples.
And Gerald.
I'm sitting here for the microbiome biobank. This is an enabling capability to facilitate research in the interface between microbiome, human health, and it's not just the refrigerator with receptionists in front of it. It's integrated with the capturing and analysis of as many phenotype data of patients which we see. And it's an ongoing process. We consume actually biomaterial, but it supports many consecutive studies in the field of inflammatory bowel disease, in the field of neurogastroenterology, but equally in the field of hepatology or inflammatory diseases of the liver.
Okay. Are there any questions from the audience at all? Graham.
So this is probably a question of anyone in the panel. How much are you looking at Indigenous health, and in data space, we're looking a lot at, we've talked a lot about FAIR, but now having to talk about the care principles. Does that apply to, well, I assume it does. How do you manage that in a tissue banking environment?
So really, as far as biological samples go that we've collected from research participants, they have the right to reclaim those samples if they wish, and we've given them that option. That includes Indigenous participants who often only have specific samples that they will donate and for specific reasons, so that has to be accounted for. There's a consultative process that we go through, both with Indigenous participants and our other participants, to gauge what they want done with their samples. It is quite complex though, because there are multiple questions around the samples that we collect. So we attempt to maintain information and contact with participants through newsletters as well, so that they have that connection maintained with us, with the intention that if they change their mind about participating or the use of their samples, they can reclaim them.
Glenda.
We have, with the clinical database for chronic kidney disease, we have a lot of Indigenous patients because of the high level of chronic kidney disease in the Aboriginal population. When we started to collect the samples, we didn't have, and we still do not have, a question in the PICF about the status of the patients as far as their Indigenous status goes. It's something to really think about. It wasn't, we did consider it. I was going to say it wasn't considered. We did consider it. And we decided that we wouldn't put a statement in there, that we just basically would take the patients who came into Site A that I talked about, where we had a lot of success, and there are Indigenous patients there. So I don't know the answer for that, but certainly they can retrieve, anybody can retrieve samples if they so desire. So that's in the patient consent form.
There's a question.
It's an integrated approach. We're not differentiating, but we have the information available, whether it's a First Nation person or not. And we have no dedicated Indigenous First Nation projects, because the administrative approval barriers probably are prohibitive at this stage.
There's a question that's come through online from Erin Davis, and Erin asks the panel, what's their view on how biobanks should be funded? Should they be funded by the users of the biobank? In other words, the research studies themselves, federal government funding, state government funding, philanthropy. Andy, can I pick on you and just ask your thoughts on that, and then we'll throw to the rest of the panel.
Yeah, sure. Thanks, John. Look, we've been really fortunate in that we've had long-term and stable funding from the Children's Hospital Foundation, which is a philanthropic source, obviously. My personal view for a long time is we've never pursued a cost recovery model for very good reason. One is that you simply don't recover cost. And I think having led lab-based and clinical research projects myself, there's enough expense in terms of reagents and everything else, staff, etc. We do get, having said that, we do get our researchers to pay for shipping, but the samples themselves are free. But we do ask that the researchers pay for shipping, and that's where our network approach comes into play. If we can couple samples from multiple sites in Australia and then do one shipping, particularly if it's, you know, shipping on dry ice, as people will know, is heinously expensive if you're doing that overseas. So you only want to do that once if you can. I think there is a role. We very clearly provide a value add to the clinical service at the hospital. So I think there probably is a role for government funding of biobank activity. We're really, as evidenced by my talk, it's a very close three-way collaboration and partnership between the clinical service, Pathology Queensland, and UQ as the academic sort of host. So I think there is a funding model to be looked at there at a government level as well, particularly if you're, you know, a key enabler and facilitator of clinical services.
So Andy, I can appreciate that you don't charge fees if it's something within the Children's Hospital or within the Paediatric Oncology group across Australia. But what about if you get approached by a third party, someone that you're not actually actively collaborating with? Do you charge those sort of arrangements a fee?
No. So the vast majority of our collaborations are ones that we have sought out, and we've taken a push out approach rather than a shop front advertise and they will come. We do get some inquiries, but the majority of our collaborations are ones that we have identified or our clinical partners have identified because they've come back from a conference and they, you know that they've introduced themselves or they've got a connection with Professor Bloggs at, you know, at an international site, and often, you know, there's a call out to say we're looking at this. People know it's a rare cancer. We'll come back. Yeah, we've had a handful of those over the years. What have we got in our tumour bank? And we reach out to say, look, we've got 6 of these samples. Would you be interested? And of course, the answer is always yes. So that's kind of been our approach, and that really underpins the collaborative model that we've had. The researcher generally has some budget for the shipping cost, but we don't charge them for sample cost recovery because you cannot put a figure on it. I know there are different schools of thought on that, but that's been our approach, and the foundation have been very receptive to that because, you know, it's already been funded. Those samples have already been funded.
What about the panel? Do people on the panel take a similar view, or do you take a different? Gerald?
I think you need to be crystal clear about the value proposition of your biobank. And the value proposition is, I mean, something which was just mentioned, rare cancers, and you collect over time volume that you can do reasonable search. But at the same time, for our models, is engagement with pharmaceutical industry who are interested in materials, interested in the data, and by having a unique selling point, you engage external parties. And while we had initially some funding from the PA Foundation, we have now been quite successful to attract MRFF, NHMRC funding to collaborate with researchers within the Queensland environment who then attracted funding. And this helps us to support the biobank. Therefore, it's a value proposition. We're not expecting to get a foundation to fund forever, but we need to be agile to work on our value proposition to continue to attract the funding.
Okay.
There was a few people who will recall a group that was set up, I think, by PA, but it was, may not have been PA, but it was all interested parties in biobanks. And the discussions finally came around to having some sort of, because of the costs of the freezers and the risk of losing very valuable specimens if they're just local freezers without backup. We're lucky at TRI, we do have backup if the freezers go down. And they talked about some sort of central infrastructure, but it was just too unwieldy, and I think they gave up on it in the end. It was a huge cost, and it was who owned the tissue once it got into this central biobank, and who would pay for the people to take samples out. And it was, I think my personal feeling is we should just have our local biobanks that are well protected, but that we don't start some sort of really huge central biobanking process.
Thank you, Glenda. I just want to ask a question about consumer engagement. Can you hear me?
Just.
Yeah, okay, but we'll come to that in a minute. Thanks. Can you hear me now?
Yep.
Okay, I just want to ask a question about consumer engagement. I mean, we've heard a little bit about this in some of the presentations and the governance, but how do you ensure that consumer engagement is real and not token? Do you involve them in the governing board, or is there some mechanisms that you can recommend? I just throw it open to all of you. Rebecca, would you like to start?
In our particular situation, with both collections that we have a steering committee, and that is a very broad collection of people and their input in terms of, we have researchers, we have clinicians, we have the pathologists. It's engaging all the possible pathways that a patient will follow through. In terms of what researchers want, in terms of, samples, data, it's very important to make those connections up front. Otherwise, you're collecting samples that really may not align between clinical pathways and research pathways.
I just note that Andy has to leave for another commitment. We've just got a couple. So thanks very much, Andy, for your contribution. We've still got one or two questions to deal with. So we might say goodbye to you, and we'll see you again some other time. Right. I noticed that Vanessa's got a question just back here. Do you want to?
Thank you.
Thank you. On the mainly biobanking is at the lab level, at the institute level, at the hospital level. Before COVID, I've been part of some workshop for a national initiative for biobanking. And I would like to have your idea of if at any time, like in the next couple of years, I hope, we will in Australia have some kind of program. In other countries like Canada or Denmark, they have a national program where actually you have an opt-out option, but it's compulsory for any kind of people working into an hospital, and they will just donate whatever gets cut from their body. And that is really helping for research on a national basis or even on an international basis. So I would like to know if you think that that would be something that Australia could look forward to, and if we have any chance of that happening.
Vicki.
So I do know that locally you can have those options put in place through your ethics committees, but not nationally. And it has been a minefield. We've started to think about that in relation to the collection of placentas, and they have the opt-out option at some hospitals around Australia, but not at others. And it is definitely something that would be a lot easier in terms of collection of tissues that often get thrown away.
I think to even further that Vicki has just touched on, that even before that step, where there's an opt-in or an opt-out to even be contacted by researchers, that has been quite a minefield in itself, and is still, there's certain sites that still struggle with that process. So to then take that one step further and talk about tissue sampling, I think is a little further away than just the next few years.
Yeah. I mean, your question actually goes back to the consumer engagement. Because if we ask our patients, our consumers, and we do this in a very systematic clinical way where we do mirror meetings, we have groups of patients who talk to our staff and they don't see the staff, and we talk about the service experience, but also involvement in research. Virtually all patients are very much in support to be involved, to donate, to be actively in this. And I think an opt-out would be a really good option as a regulatory approach, but to have an administrative process which then regulates everything, what goes into the biobank, what question, what data, I think we would lose the agility which we need to be competitive on a national and international schemes. Therefore, the small biobanks give the advantage that we can respond to trends and being very successful with NHMRC and MRFF. If it's a national initiative, you have the smallest common denominator, which probably will not serve the purpose.
Okay, perhaps if we can move on and make this the last question, Diffu. Thank you.
For this very informative discussion, I have a question around the selection criteria for the samples included in the biobank. In the beginning of the setting of biobank, there's a certain scope for project, but there may be other research direction and will be added and follow after the biobank has been established. So how you decide how broad or how narrow is setting the selection criteria for the sample, including the biobank? That may have to consider how much resource and the capacity of the biobank. So that's my question.
Vicki, do you want to make a quick comment?
Yeah.
And then we'll wrap it up.
Okay.
Thanks.
We had a consultation process across 50 institutions and 150 researchers to ask what samples they wanted and what were the topics that they thought were important for the Queensland family cohort. That then fed into our ethics application. And now everybody who's interested in accessing those samples provides a sub-study application to ethics, which comes under one of the 22 themes, of which they then get approval to access the samples and data. So we established it right at the beginning, which is a better way to go in terms of allowing people to access the samples in the future.
Okay, thank you. Thanks to all the panel. Thanks to Glenda and Andy, who's now left, and thanks to the audience for your questions. I think it's been really useful just to hear about some of the variety of biobanking that's going on and some of the issues that people have faced, but also the rewards and the great things that have come about as a consequence of those biobanks. So thanks everyone for your attention. We look forward to seeing you at the next TRI seminar.